Hi everyone, I'm Xiao, a PhD student from CSBL. Today my presentation title for the journal club is High-throughput genetic engineering of nonmodel and undomesticated bacteria via iterative site-specific genome integration. Here are the highlights of the article. This article describes the serum-assisted genome engineering, an easy-to-use, highly efficient, and extensible technology that enables selection marker-free, site-specific genome integration of up to 10 DNA constructs, often with efficiency on par with or superior to replicating plasmids. SAGE uses no replicating plasmids and thus lacks the host range limitations of other genome engineering technologies. Researchers demonstrate the value of SAGE by characterizing genome integration efficiency in 5 bacteria that span multiple taxonomy groups and biotechnology applications. Additionally, they have identified over 95 different heterologous promoters in each host that consistently drive transcription across different environmental conditions and genetic contexts. This breakthrough highlights the significant potential of SAGE in rapidly expanding the range of industrial and environmental bacteria that can be utilized in high-throughput genetics and synthetic biology applications. To address various societal challenges, the application of synthetic biology often faces limitations due to the lack of effective genetic tools, especially when working with microbes that are intended to be deployed in environments where antibiotic selection is undesirable and impractical. The commonly employed methods for transferring foreign DNA into bacteria, such as replicating plasmids, homologous recombination-based allelic exchange, and transposon mutagenesis, have inherent limitations that restrict their usefulness in studying engineered microbes. Replicating plasmids are often not maintained stable or come with fitness costs, necessitating constant selection, typically through antibiotic resistance. While transposon and homologous recombination-based technologies enable stable integration of foreign DNA into the genome, they are generally incompatible with high-throughput rational engineering studies. The integration sites resulting from most transposon-based tools are unpredictable, which can lead to inconsistent host pathway interactions among different transformed strains, making reliable comparisons between pathway variants challenging. Homologous recombination-based allelic exchange, regardless of the counter-selection method used, is typically inefficient, making it difficult to construct even moderately sized strain variant libraries. Phage integrase-mediated recombination technologies provide the scientific community solutions to many limitations stemming from common genetic tools. Phage integrases, also known as site-specific recombinases, are enzymes that catalyze recombination between 2 specific sequences of DNA. And here is the mechanism of the site-specific recombination. In any conservative site-specific recombination event, there are 8 chemical steps: strand, cleavages, and 4 ligations. Cleavage occurs when a nucleophilic amino acid functional group at the recombinase active site attacks the cis phosphodiester bond of a DNA strand. The immediate product of cleavage has a broken DNA strand, with a covalent phosphodiester linkage between one DNA end and recombinase at the breakpoint. Serine recombinases become linked to the 5' end of the DNA, leaving a 3' hydroxyl group on the other end at the break. Serine recombinases cleave all 4 DNA strands in the synaptic complex, creating double-strand breaks at the center of each crossover site. Each half site thus formed has a recombinase subunit covalently attached to its 5' end, and second single-stranded protrusions terminated by a 3' hydroxyl group. The half site are then exchanged and religate, creating recombinates. This mechanism contrasts with that of the tyrosine recombinases, which become linked to the 3' end of the DNA at the strand break and do not make double-strand break intermediates. Instead, they cleave, exchange, and religate pairs of single DNA strands. Thus, strand exchange proceeds via a four-way junction intermediate with two recombinant and two non-recombinant. Tyrosine recombinases leave identical or highly similar scars that flank each DNA insertion. Sequence similarity between scars causes genetic instability. For example, inserted DNA can be excised by homologous recombination, and reversibility precludes their reuse for sequential insertions. On the contrast, serine recombinase follows a unidirectional pathway, resulting in the creation of two new attachment sites, ATTL and ATTR, that cannot undergo recombination without the assistance of accessory proteins. Additionally, serine recombinases do not rely on host proteins and have been widely utilized in various organisms, indicating their potential applicability across different species. However, existing recombinase-based tools often have limitations, such as being capable of integrating only one or two DNA fragments, functioning within a restricted range of host organisms, relying on plasmids that replicate within the host, or frequently leaving selection markers within the host chromosomes. To overcome these challenges and the limitations, researchers focus on the development of a toolkit called Serine Recombinases Assisted Genome Engineering, SAGE. This toolkit utilizes highly efficient serine recombinases, which are transiently expressed from non-replicating plasmids. The SAGE toolkit enables the iterative integration of up to 10 different DNA constructs, or variant libraries into various bacterial genomes at specific ATTB sites, as shown in figure. This schematic depicts the SAGE cycle for site-specific integration of multiple DNA fragments into bacterial chromosomes. To construct a base strain, ATTB sequences are integrated into target microbe's genome using standard methods like allelic exchange or transposase integration. Next, one of the system's 10 recombinases is transiently expressed from a non-replicating helper plasmid, catalyzing recombination between its cognate ATTP and ATTB sequences located on a co-transformed target plasmid and the host chromosome, respectively. Recombination integrates the ATTP target plasmids into genome unidirectionally, generating ATTL and ATTR sequences to excise unneeded plasmid features and prime the strain for additional SAGE cycles. Pi C31 integrase is transiently expressed by transforming a non-replicating backbone excision plasmid. Pi C31 integrase catalyzes recombination between Pi C31 ATTP and ATTB sites of the integrated target plasmid, excising the selection marker and the E. coli origin of replication from the chromosomes. For organisms with faster growth rates, the SAGE process, which involves integrating ATTP target plasmid DNA into the genome and subsequently excising the plasmid backbone, including the selection marker, can be completed in as few as 8 full workdays, as shown in the figure. As shown in the table, researchers selected 5 bacteria that represent a range of biological diversity and varied application spaces to demonstrate SAGE. Specifically, they tested performance with the domesticated microbes, Pseudomonas fluorescens SBW25, Rhodopseudomonas palustris CGA009, and Pseudomonas putida GP01. Domesticated bacteria indicates that bacteria adapted over time as by selective breeding from a wild or natural state to life in close association with and to the benefit of humans. They also tested SAGE with both a model actinomycetes, Rhodococcus josti RHA1, and an undomesticated Pseudomonas that they isolated from the endosphere of sorghum bicolor roots under drought conditions. Here is the SAGE cycle designed for this strain in this paper. First, they prepared the SAGE-compatible base strains by integrating a poly-ATTP cassette into genomes of each organism. The cassette contains an array of 10 distinct ATTP sequences, each separated by a unique spacer sequence to enable polymerase chain reaction screening for plasmid integration. And the array is flanked by double RHO-independent terminators that should provide transcriptional insulation for many bacteria. To integrate poly-ATTP sequence into target microbe's genome, researchers used 2 different methods in this study. For Pseudomonas fluorescens, Rhodopseudomonas palustris, and Rhodococcus josti, standard homologous recombination-based allelic exchange methods were used. Allelic exchange is defined as the replacement of a specific region of DNA by a homologous recombination. These replacements can take many forms, with the most common being replacement with an antibiotic resistance cassette, a complete deletion of a region of the chromosome. Homologous recombination occurs between the DNA in the upstream regions or the downstream regions of the target gene and the recombinant plasmid, resulting in the integration of the plasmid into chromosome. A second recombination event can either occur between the upstream regions or the downstream region, resulting in the regeneration of the wild-type allele or the formation of the mutant allelic exchange construct. Similar to many undomesticated organisms of interest, other 2 strains lacked sequenced genomes as they initiated this work. They introduced the cassette into 2 hosts through electroporation of an TM5 transposon complex. TM5 transposase is a bacterial enzyme that integrates a DNA fragment into genomic DNA. In the first step, individual molecules of transposase bind to the specific site at the end of the transposon DNA. In the next step, looping of the transposon DNA results in formation of a synaptic complex that brings the 2 ends of the transposable element close together. Once the synaptic complex has been formed, the TM5 transposase cuts the transposon DNA away from the flanking donor DNA. After cleavage, the TM5 transposase can move about freely until it encounters and binds to the target DNA. Through a process called strand transfer, the transposase catalyzes insertion of the transposon DNA into the target DNA, completing the transposition process. To enable the reuse of the selectable marker in SAGE, it is necessary to remove the marker from these strains. For this purpose, they designed the transposon in a way that allows easy removal of the antibiotic resistance gene MBT2. They incorporated a pair of pi C31 ATT BMP sites flanking the MPT2 gene. Consequently, transient expression of pi C31 integrase leads to the excision of the MPT2 marker from the genome. To achieve this, they introduced a non-replicating plasmid that expresses pi C31 integrase into each strain containing the TN5 integrated poly-ATTB cassettes. The fact that marker-free strains could be identified without the need for selection highlights both the exceptional high transformation efficiency of these strains and the effectiveness of site-specific recombination. They utilized these resulting NPT2 knockout colonies for each organism to evaluate the performance of SAGE. They evaluated the functionality of each recombinase through a plasmid transformation assay. In this assay, all 5 organisms were simultaneously electroporated with 2 plasmids. The first plasmid was a non-replicating helper plasmid, PGW31 to PGW40, that expresses a specific SAGE recombinases. The second plasmid, called PGW60, was a non-replicating target plasmid containing a series of cognate ATTP sequences for each recombinase, along with a constitutively expressed neon green fluorescent reporter. The transformation of PGW60 plasmid was expected to occur through recombinase-mediated recombination between the ATTB site on the chromosome and the corresponding ATTP site on the plasmid. To serve as parallel controls, they also assessed the transformation efficiency of replicating plasmids based on PBBR1 and integrative factor PST152 in the Proteobacteria and Rhodococcus josti RHA1 respectively. The PBBR1-based plasmids, namely PJE354 and PEYF2K, contained reporters such as amnion green and MKT2 respectively. Transformation efficiency was measured by enumerating colonies' resistance to kanamycin or apramycin. Unexpectedly, Rhodosudomonas palastres were unable to transform with PJE354, which could be a consequence of poor MPT2 expression or plasmid instability. While the performance of recombinases varied among different organisms, at least 8 of them effectively integrated PGW60 into the chromosomes of all tested organisms. Among the 4 proteobacteria, co-electroporation of BXB1, TG1, R4, or BL3 helper plasmids with the poly-ATTP plasmid PGW60 resulted in transformation efficiencies comparable to or even better than the replicating control plasmid. These recombinases also performed well in Rhodococcus josti RHA1, although the efficiency was slightly lower than that of the control plasmid PSET152. This could be attributed to the fact that SAGE requires simultaneous transformation of 2 plasmids, unlike control plasmid only requires 1 plasmid. Alternatively, the lower efficiency could be due to suboptimal optimization of recombinase expression and selection marker cassette for actinomycetes. The pi C1 integrase generally showed lower efficiency, possibly indicating inadequate expression or the need for a more stringent operating environment compared to other recombinases. In each organism, At least 4 out of the remaining 5 recombinases demonstrated high transformation efficiency, although in all hosts except Rhodopseudomonas plastis CGA009, the transformation efficiencies were lower than those observed with replicating plasmid controls. Importantly, no colonies were observed in any of the 5 hosts when PGW60 was electroporated without a recombinase helper plasmid. And serine recombinases generally have a preference for their native ATT sites, but they can also target other sites in the host chromosome known as pseudo-ATT sites. Therefore, integration accuracy is an important factor when evaluating the suitability of each recombinase as a genetic tool. To assess integration accuracy, they conducted a colony PCR assay using specific primers for each organism to determine the frequency of PGW60 integration within the poly-ATTB cassette compared to other chromosome sites. Generally, recombinases that exhibited high transformation efficiencies in a particular organism also demonstrated high accuracy in that organism. BXB1 showed the highest accuracy with 100% accuracy in all 5 organisms. RVTG1 and R4 also exhibited high accuracy with 100% accuracy in 4 out of the 5 organisms and around 50% accuracy in the 5th organism. Although pseudo-ATTP sites were not identified in the 5th organism, the approximately 50% accuracy suggests the presence of another ATTP site in the genome with similar affinity for the recombinase. On the other hand, the BL3 integrase consistently displayed high but imperfect accuracy in all 5 organisms. This may indicate a lower fidelity recombinase or the potential for improving the ATTP sequence through ATTP site engineering. The integration accuracy of the remaining recombinases varied significantly between organisms, but they demonstrated reasonable accuracy in some of the organisms tested. Variation in accuracy between organisms could be attributed to native sequences in the host genome serving as alternative ATTP sequences, which may even have higher affinity for the recombinase than the ATTP sequences present in the engineered poly-ATTP cassette. Therefore, the presence of pseudo-ATT sites depends on the chromosome sequence, and integration accuracy must be evaluated independently for each host. To reuse the selection markers for SAGE cycles and for strain deployment in many operating environments, the selection markers designed in SAGE cycle were removable. So, as shown in previous figure, all ATTP target plasmids were designed such that the cargo region was flanked with a cognate pair of pi-C31 ATT sites. To confirm the plasmid excision in bacteria, the SECB selectable marker was incorporated into ATTP target plasmid backbone, which allowed for the selection of plasmid excision by culturing the bacteria in the presence of sucrose. A modified version of PGW60 containing SECB, called PJE1818, was created and used along with BXB1 integrase to integrate the plasmid into genomes of Pseudomonas fluorescent JE4621 and the Rhodopseudomonas plastis JE4632. The resulting strains became sucrose-sensitive and were unable to grow in the presence of 25% and 5% sucrose. Each strain was then transformed into 2 plasmids, non-replicating pi-C31 integrase expression helper plasmid called PJE1817 and the plasmid containing a temperature-sensitive origin specific to Pseudomonas called PGW30. The transformed strains were cultivated on agar media containing sucrose to select for cells that have excised the PGE 1818 backbone. Among the sucrose-resistant colonies, all Pseudomonas fluorescens PGE 1817 and 90% of Rhodopseudomonas palustris PGE 1817 transformants, as well as or PGW30 transformants, were sensitive to kanamycin, indicating successful marker removal. The excision of the plasmid backbone was further confirmed through PCR analysis in colonies that showed kanamycin sensitivity. Researchers emphasized that the genetic stability in the absence of selection through antibiotics or orthotrophy is the critical advantage of the SAGE-mediated DNA integration compared with replication plasmids. And they proved it by comparing the stability of fluorescent protein expression cassettes maintained on replication plasmids or SAGE-integrated genomes. As a result, fluorescent reporter expression was substantially more stable in strains generated by SAGE than strains generated by transformation of replicating plasmids. As shown in figure, flow cytometry measurement of fluorescence in population of Pseudomonas fluorescens JE4621 containing no heterologous expression construct labeled as parent strain, a replication neon green expression vector PJE354 or non-replicating neon green vector that has been chromosomally integrated. Except parent strain, the starter culture were cultivated in medium containing kanamycin sulfate for plasmid selection. The x-axis indicates the relative fluorescence of each cell. The y-axis represents the abundance of cells in the population with a given relative fluorescence unit. Expression was stable in all strains generated using SAGE, with larger than 99.75% of cells expressing neon green after 20 generations of growth without selection in Pseudomonas fluorescens. However, replicating plasmids were lost rapidly without selection. Only 2.5% of Pseudomonas fluorescens and populations produced fluorescent proteins after the multipassage cultivation in the absence of selection. In addition, the presence of multiple SAGE-integrated excision scars does not impair genetic stability. Cis plasmid backbone, when removed, create a 47 base pair pi C31 ATTL sequence scar in the chromosome. The existence of multiple pi C31 ATTL sites could potentially cause genetic instability due to homologous recombination. So they evaluated the stability of neon green and MK2 expression in Pseudomonas fluorescens JE5117 recombination between the two pi C31 ATTL sequences in the strain would lead to excision of the MK2 genes and the loss of red fluorescence. They cultivated two lineage of JE5117 and used flow cytometry to measure the fraction of the population that displayed both red and green fluorescence after 100 generations. In both lineages, the fractions of the population that displayed both red and green fluorescence remained steady. The development of genetic components such as promoters, ribosomal binding sites, and terminators is crucial for controlling the expression of foreign genes in bacteria. Achieving a balance between engineered gene expression and essential cellular functions is important to minimize the impact on host fitness and ensure long-term stability. Furthermore, bacteria are often exposed to changing environments, highlighting the need for promoters that can consistently drive gene expression under diverse conditions. To adjust this, they have created and validated a methodology to evaluate extensive libraries of genome-integrated promoters across different environmental conditions using SAGE-capable host organisms. To evaluate a collection of 287 synthetic and natural promoters integrated into chromosomes, the researchers employed a modified version of the high-throughput sequencing transcriptional activity assay introduced by Jones. This methodology combined the efficiency of SAGE integration with certain adaptations. Instead of using a single barcode per promoter, they utilized 5 distinct barcode variants for each promoter, totaling 1,435 promoter and barcode variants. This allows for technical replicate within the experiment, enhancing data reliability and identifying and eliminating outlier barcode variants. Additionally, they positioned the barcode within the 5' untranslated region UTR immediately upstream of the RBS rather than within the coding sequence of the reported gene. This placement facilitated analysis of protein sensitivity to variants in the 5' UTR sequence, an important factor affecting transcript abundance. To identify promoters with consistent expression across different 5' UTR sequences and environmental conditions, a pooled library of barcoded reported plasmids was integrated into the genomes of the 5 organisms using SAGE. Mean expression statistics were used to assess promoter sensitivity to genetic and environmental contexts. Promoters meeting the criteria, while the mean RTA, relative transcriptional activity, of each non-outlier barcode variant fell within a 3-fold range for 5' UTR sequence variation and within a 4-fold range for environmental conditions, were classified as insensitive to those factors. Promoters meeting both thresholds were labeled as consistent promoters. The accuracy of RTP quantified expression was validated by comparing barcode sequencing results with plate reader-based fluorescent assays, specifically for a subset of related promoters present in the Pseudomonas fluorescens library. The expression levels measured using both methods exhibited a strong correlation, suggesting that high-throughput barcode sequencing provides comparable results to fluorescent protein reporter assays. And strain-to-strain expression level correlations decreased with phylogenetic distance. When calculating correlations for only the consistent promoters, they found correlations increased for most comparisons, especially for Rhodos-Pseudomonas palustris correlations to the other proteobacteria. And here is the conclusion for this article. The SAGE toolkit extends the accessibility of high-throughput engineering for bacteria in any application space. Also, the toolkit includes non-replicating and conditionally replicating plasmids for recombinase expression and ATTP-containing target plasmids with multiple options for selection and counter-selection markers. DNA integration by serine recombinases should be selectively reversible by co-expression of a recombinase with its cognate recombination directionality factor protein. And SAGE is orthogonal to host recombination machinery and does not require use of any replicating plasmids and thus is theoretically extensible to any transformal bacteria. Thank you for your listening.